I realized that I had forgotten to go over that. So, okay. So for the view slides, so for the drug development, so with that, so you're going to need to know, so for that, so what KD stands for, and it's basically the concentration where 50% of the receptor is bound. Okay, it's similar to like how we would calculate Km, and that so for that, so when is at what concentration it does, and it's given as a concentration, so micromolar, nanomolar, picomolar, or whatever, it's given as a concentration. So if you don't have a receptor, you can look at like a living system, and so that you have EC, which is effective concentration. So where you kill the cells. So if you're looking at a whole, the living system, does it survive? Does it not? At what concentration do you get that biological response? The other one is inhibitory concentration. So at what point is half of the signal lost? Things like that. So if you're looking at, if you're trying to suppress expression of a protein or a receptor or whatever it is, you don't need to know this calculation. What you just need to know that IC, again, we're looking at the 50% mark. At what's the concentration where half of whatever is inhibited. So and the lower the better. So know what ADME stands for, right? It's absorption, distribution, metabolism, excretion. Okay, so you have to know what that means. Okay, so so absorption is just where does the drug go, or you could say, I should say, how that's actually distribution. How well does the drug get out of the pill after you swallow it? So how well does it get into your body? Okay, this is what's sometimes called oral bioavailability, and you do need to know Lipinski's rule of 5, and so this is what this is that sort of general sort of rule of thumb with that on drugs and on what makes things bioavailable. Okay, and so you need to know you need to know these in general, right? So you don't want a molecular weight generally above 500, more than 5 hydrogen bond donors, 10 acceptors, or log P of greater than 5. So remember, log P is the concentration of the drug in octane versus water. Okay, so P is the concentration of your drug in octane versus water, drug in water. So and then you take the log just to make the number look good. It's an easier number to do. Stuff like that. And so if this is 10,000 to 1, okay, so this would be the log of that, right, would be 4. So that means 10. So a log of 4 would be 10,000 molecules in the octane for 1 water. So there's not very much soluble actually in the bloodstream. Okay, it's going to want to attach to something hydrophobic. So it's a measure of how greasy the molecule is. Okay, so with that, and so so if you're coming in here and and so just have a good sense for these. Okay, so distribution is just where does the drug go within the body. So pull this down. Hopefully a little bit easier. Let me pull this down a little bit. Maybe it'll be a bit easier to see. Okay. So where does it go? Where does the drug go in the body? So that's the distribution part. Metabolism. So how does the body break that down? Okay. And it does it primarily either by oxidizing it, by making it making H's into OH's to improve the solubility, or conjugating it, so adding other soluble molecules onto it. It's like glutathione, sugars, sulfates, phosphates, things like that, stuff like that. So, so the way that it breaks it down is it actually reacts with the molecule in order to be able to get it more soluble so that you can excrete it. Okay. So, how quickly that it breaks down is going to be really important, right? So for that, because after you take the pill, right, you're gonna get this rise in concentration within the blood plasma, so for that, and then your body's going to slowly degrade it. Okay, and so they know that how the rates of these curves, right, depend on the will be dependent. The rates of this curve here makes it such that so they can figure out the drug dosing. Okay, and so they assume that this is what happens, and so in order to be able to maintain a minimum level, right, they need to know how quickly your body gets rid of it. So if you inhibit that, okay, if you inhibit that process, this metabolic process, right, it'll slow this down, and so when you take another dose, you can get up to toxic levels. So you have to be careful with that. So excretion, right? So with that, so how does the body get rid of the drug? And there's two primary ways: it's urine and feces. Okay, so you have to, so they measure, so they collect it and measure it. Okay. So that's the ADME. Now, one of the things you have to be careful of, right? You got to get the dosage correct, because if you don't, it can become toxic. Okay, so if the metabolism is off, you can build these up and to get into toxic metabolites. Okay, so either off-target effects or metabolites would be this. And so you want to know what the therapeutic index is, the ratio of a lethal dose versus an effective dose. And so what you want is as large of it, you want this gap to be as high as possible. So you want a really high therapeutic index. Okay, so for something like this, right, you want something, you'd want the drug sort of over in here. So 1,000 to 1. So, if you took a really high, if you accidentally took a dose that's 10 times higher, you'll still be okay. So with that, so you want this for safety concerns, particularly things that for chronic conditions. You don't want to accidentally, if your metabolism gets screwed up a little bit, you don't want this to be, close, because, you can start building this stuff up. You don't need to know this chart, but just whatever the therapeutic index is. Okay. Next up, so for that, so know the major routes of the drug discovery, not the actual drugs themselves, but how we get there. So it's serendipity. You notice something and you figure out, something's happening that's weird. You figure out why. Natural products can be a source of new compounds. So with that, so combinatorial chemistry and high throughput screening. So this is just throw a lot of compounds in it, make a bunch of different compounds, and do a lot of assays with that, and see if you can find ones that hit, and then you can design your drug from there. And then the last one is what's called structure guided design. And so it's this combination of, we have a lot of information that we can use to design the drug, to design some candidates. And so let's do, instead of 10,000 that we did with the combinatorial chemistry and high throughput screening, let's do 100 compounds that are much more targeted, so we can get much better information on it. So those are the sort of 4 primary ways of drug development. Okay. So genome analysis. Okay, so with that, so so if you guys hadn't, so there was actually a Ken Burns. So keep an eye on it for if they show this again. Ken Burns, the guy that does a bunch of documentaries on PBS, he did one on the gene. Something like that. So the evolution of sort of the discovery of DNA and what we've done with it. He had a 2-part series. It finished up. What was it? It finished up this week. I forgot to mention that to you guys, but it was a really, really cool series. So if you get a chance to watch it, please do. Something like that. So if I find it, if I hear that it's on again, they replay it, I'll send out the link. With that. So, okay. But one of the things that comes out of there, particularly for like the Human Genome Project and the other sort of genomes that have been other organisms, what you can do is you can compare not only the sick, within human populations, those that have, are sick versus those that aren't. With that, so you can compare the two and see if can you find the differences, and those differences may hint at, hey, if we go after this particular target, right? So that we might be able to we might be able to a lead for treating that condition. Okay. The other thing that you could do is if you're looking at a pathogen versus host, right? So if you compare those genomes, are there unique pathways within the pathogen that would allow you to target that just the pathogen without hurting the host? So particularly things like viruses or bacteria, those have unique pathways that aren't in mammalian cells, and so we can go after those. Okay, so animal testing is the is the primary way before it gets into humans. You can do these functional testing of drugs. You can knock out different. You can do studies with them where you're looking at, is this target a potential, potential to change, to modify a drug, so you can knock the gene out. So remove it from the genome and see what happens. You can also then dose the mice with drugs and see how they work. There's also some toxicological studies that need to be done, particularly which the FDA require to make sure that we don't make toxic metabolites. We don't want to treat for high blood pressure and destroy your liver, right? It's not going to do any good. So you need to be able to go in there, test them out, collect them, find out how the drugs are metabolized, and then work on, to make sure everything's okay before you get it into humans. Again, so this is so you talk about pathogen-specific pathways. So this one just so happens to not be the SARS. This is SARS-CoV-1, where now we're dealing with SARS-CoV-2. So a little bit different protease. So okay, so you're gonna need to know the drug phases 1, 2, 3, 4, and what you're looking for primarily is what's the goal of the phase, right? What is it that they're looking at in phase 1, phase 2, phase 3, phase 4? And then what's the number and the type of people tested, right? So is it a small number of healthy people, small number of sick people, large number of healthy people, large number of sick people? With that, so how many and what cohort of people are we looking at? Now, he doesn't mean like death's door type people, right? But it's people that have a disease, right? So people that have, if you're looking for a diabetes drug, right? So you would do, for phase 3, you would do a large number of people that are sick with diabetes, with that, or for cancer drug, people that have cancer, right? Things like that. So that's what you need to know for each phase. Okay. So for phase 1, this is all about safety. So with that, so you're going to give a sub dose to determine safety profiles in humans. This is the first time it's going to be first in human. So with that, so make sure everything's okay with it. And it's a small number of healthy volunteers. That go in there and they sublethal dose or sub-therapeutic dose. Hopefully it's sublethal. So with that, just test for safety. Okay. Phase 2, this is the first time it gets into sick patients. So these are the patients with the disease that you're looking at. It's a small number, and it's two things. One is to see this proof of concept. Does the drug appear to work? Does it improve their condition when it's combined with another therapy or therapy alone? So with that, and then it's determined, but then you're also looking at the proper dose size, right? So if you're looking at, say, 3 doses, is the large, is there a dose dependence response? And if it is, what's the most appropriate dose, right? So if, if you go from 10, 20 to 40 milligrams, right? So with that, and so 10, 20 is good is much better than 10, but 20 is, but 40 works about the same as 20. Then maybe 20 will be your lead dose, stuff like that. So you need to know that, or like the drug company would need to know that, to move on. Once it gets approved for phase 3, now it's the large actual scientific thing. So you're going to need a large number of sick patients, patients with that condition that you're looking at, and it's placebo controlled. So you're going to get, unless there's no treatment for it, this is going to get added onto the existing treatment. So normal treatment plus your new drug, or normal treatment plus a placebo. Okay, so placebo is just a sugar pill. Okay, but it looks exactly like the regular drug. Okay, so for that, but neither the patient nor the doctor is going to know who is in which group. Okay, and stuff like that. So that there's no this sort of bias. Saying, oh well, we know that this is the the placebo, so don't worry about it. It's no big deal. It's like that. We don't want that sort of that bias in the drugs. So you don't know. The data is sent back to the pharma company, and then they can analyze to figure it out. So that's what they mean by double blind. Both the patients are blind and the doctors are blind to who is in what group. So about that. So they're typically long, right? So about that, because you need to get good statistics on and know about toxic side effects, or if it actually is working. Okay? Phase 4 this is usually a large, the overall population, so it's a large number of sick people again. But this one it's looking for any potential toxic side effects within the larger, human population. Okay, so keep an eye open. You can only have they're not going to look at everybody in the phase 3 trial. You're only going to look at a small subset. So now you're opening it up to the entire population, and we need to keep an eye on it. So on the drug side effects. So drugs can be resistant to this, can become resistant over time. And the classic ones are the antibiotics, antivirals, and anti-cancer. These literally have a Darwinian evolution sort of component to it. They have, these things can create mutations, particularly antibiotics and antivirals, although anti-cancer can too. So that small population of, say antivirals, you can get if there's a mutation that happens within the virus, now it's no longer resistant. Now the drug is no longer working on it, and so now that one can start to expand the population, right? So that's what's happening a lot with that's why we don't have a universal sort of anti-HIV, because it's a retrovirus. It has RNA. They mutate quickly, and so you can get the these things can stop working because they've mutated so that they don't work anymore. Antipsychiatric or psychiatric drugs, they also do sometimes, and it's the evolution of the disease, but this is a little bit more complicated. They don't exactly know why. Sometimes the drugs just stop working, and there's not a good explanation for it, because probably not a mutation, but hard to know. So something's happened, but it's a weird evolution of the disease. Perhaps, who knows what it is. So okay, I think that's the drug discovery part. Any questions before we get into the immune system? Okay. Okay, so for the immune system, right? So we have two different types, right? We've got what's called the innate immune system and the adaptive immune system. So the innate is this first line of defense. The adaptive is this antibody generating the part. So this is the more well known, but this one is this is also incredibly important. so the innate immune system works on these toll-like receptors, using these toll-like receptors, and what their job is to detect a pathogen. If it detects it, then it swallows it up in order to be able to try to destroy it. And it's really a quick response system. It's not perfect, so with that, but it does a good job. Okay, so these TLRs, the way that they're doing is not, each TLR is responsible for detecting a specific feature on a pathogen. You do not memorize this list, but just know in general, right? So like TLR3 is looking for, sometimes they're looking for double-stranded RNA, or some of them, single-stranded RNA. It's just some weird feature on this. That's how they work, is they're looking at these features that wouldn't normally be in normal human cells, or normal, eukaryotic cells, or whatever. That's what they're looking for. So so they're on the lookout. So if they see it, then they swallow it in order to be able to destroy it, and also raise the alarm to say, like, this is, we're probably under attack. So, but they just, each one is looking for it. So there's, so that's the basis of the innate immune system. There's two different parts of the adaptive immune system. One's the humoral part, and this one is looking for, this one is looking for invaders. The other one is the cellular immune response. These are the ones that are to figure out, is the cell infected? If they have an infected cell, then we're gonna destroy that cell in order to prevent the infection from spreading. Okay, so for these, we're going to use antibodies, these immunoglobulins, to mark the invaders for instruction, or for destruction, something like that. So, and what we do is the things that actually generate the antibody, okay, that start the process to create the antibody. Those are what are called antigens. They're antibody generating molecules. Okay, so these are the parts of the pathogen that start the immune response. Okay. Okay. So for this one, let's see. So you are going to need to know the general role of each one of these types of immunoglobulins. Okay. So IgG is just that's the one that's expressed is to mark the invader in order for destruction. Okay. So the IgM, this is the initial response. It's bound to the T cell. It's a pentamer, and it's this initial response one to get the ball rolling. Once it detects an antigen. Okay. Or wait, hold take that back. It's on a B cell. Excuse me. IgA. So this is the one that's in the mucus and tears, saliva, and it's trying to stop the infection before it can get in. Okay, so that's its role. IgD, we don't know. So with the IgE, it works with looking for parasites, but it's also responsible for allergic reaction. This is what usually ends up getting triggered when you have an allergic reaction to something. Okay, the IgE. Okay, so each one of these is going to have, so remember there's a constant and a variable region within these antibodies. They have they have these hypervariable loops in them. You compare them, and so on both the heavy and the light chain. Okay, so each one of these has 3 loops that are variable, and it's these loops that are what grab a hold of the antigen. Okay, if you have a small molecule, you may only interact with a couple of loops. If you have a protein, it'll probably interact with all 6, things like that. So, ooh, where did that go? Okay, so if you have a large antigen, you actually, so if you have a big virus, or you have a large, I don't know, a bacteria or something like that, it's not going to just mark one place, right? It's not going to make antibodies just for one part of that. The pathogen is going to try to get multiple ones. So even if there's a mutation that happens within that marker, you're still being able to hang on to it at a couple other places. Something like that. And so when you get a response, you're going to get it's going to make antibodies against different parts of the epitope. So okay. So okay. So when that's so if the so on the surface of the B cell, okay, so this can detect antigens, some of that. So what's happening is it has these IgMs membrane bound on the surface. Okay, if they start to cluster and bind an antigen and cluster, okay, it starts the process of activation. Okay, so it's not just one binding one antigen. It has to several of these IgMs have to come together in order to be able to bind, so that so you don't get a false positive. Okay, something weak to go by. When that happens, okay, we want the variable region to not change. Okay, we know that this variable region binds that the antigen, so we know we want to keep that. And so basically, what ends up happening is we get part of this. The part of the IgM, we cut that out, and so when we cut that out, it turns an IgM into into an IgG. Okay, so it doesn't have to completely remake the gene. It just you get a different part of it, so that, and so it's this. You end up with if you what is that called? The I can't think of the term right now, but basically, instead of these introns, getting intron switching and stuff like that, and so you can those don't get expressed, and so it's just this part here, so that gets taken out. So you're only looking at the IgG portion of it. So okay. So for this one, right? So so that's how you start the for the B cells. Okay, pathogen comes by, binds to it, activates the B cells, and so you come in there and you start making your IgGs. The other ways that we can do this is using these MHCs, so these major histocompatibility complexes, and there's two different classes. Okay, one of them is the class 1 is to see if determine if you are is to indicate whether or not you are infected. Okay, and so it displays the internal proteins on the surface. If you've been invaded by a virus, right, it's going to grab some of those viral proteins and display them out. If so if a T cell comes by and detects some of that protein on there, it's gonna mark the cell for destruction. Okay, so a cytotoxic T cell. So the T cell receptor is going to come by and scan these, right? So check everybody's badges. And so be able to go be able to make sure everything's okay. So hold on. Sorry, there's a situation with the dog, stuff like that. So we're going to mark it for destruction. Okay, so these was it the MHCs, right? So they form this groove. So with that, I always thought it looked like a taco shells. And in that groove, there's a variety, there's quite a bit of diversity in here, so it can do all sorts of different types of peptides to be able to display out onto the surface. Okay. So the part that changes this, that's looking at this are the T cell receptors. So they're sitting on the surface of these T cells, these cytotoxic T cells. Okay, they're very similar to Ig immunoglobulin proteins. They have the gut, but they are membrane bound to the killer cells, so that they don't have to get there and then similar for come. They're going to bring the whole cell with it. with that, so you can start so you can get the process going as quickly as possible. So they do get marked, so with that, so mark for destruction, so with that, using this CD8. Okay, so okay, so the other class of ones, these are on the lookout for the pathogens coming by. So it's very similar to, being able to keep an eye open, like with the B cells. But these are what are called antigen presenting cells. So with that, so there's dendritic cells are one type. They work very similarly to the class 1 MHCs, where they instead of taking your internal proteins, what ends up happening is things that swim by get grabbed and come in and get digested and then displayed on the surface. So it's actually taking proteins from the outside and displaying them in there. So it's not just it's not is giving the it's displaying the proteins from the outside as opposed to the inside. So with that, so again, T cells are going to be coming by and scanning those. For the class 1, it's looking for the infected cells. If it detects them, the cell gets destroyed. We don't want to do that with the class 2, because it would be it'd be like destroying your own troops, right? So we do get it is looking for an invader. It does start the cascade, but the cell itself is not marked for destruction. Okay. Both the MHCs class 1 and class 2 are very similar. They have the sort of the loop here that they can put things in. And here, instead of a cytotoxic T cell, it's the helper T cell that scans it. Okay. And the helper T cell, again, it's instead of CD4, it's put CD4 on instead of CD8. So we don't kill it. So this one, I think it's so again, I think this is out of place. So this one, there's a lot of diversity in here to do a whole bunch of different types. Okay, so one of the classic diseases that affects the immune system is HIV. So it targets the immune system by attacking these helper T cells. And so if you destroy that link in the process, so you can't, it prevents your ability to mount an immune response, not only to HIV, but also to other infections. And usually, what ends up happening is the AIDS is acquired immunodeficiency syndrome. What's happening is your body can't mount a defense against other pathogens, and so it's the other secondary infections that end up causing death. But by infecting the helper T cells, it prevents that itself from getting destroyed. Okay. So these T cells. So how are they selected? How does the body know what how does it know how to what is an invader and what's not? Okay, what's self versus non-self, and they go through this positive and selection, this positive selection and negative selection before they can get released from the thymus. Okay, so the positive selection here is it's saying, okay, we're going to present an MHC to it. So the initial step is, can it bind to a regular MHC? If it can, then it can move on. If not, then that T cell will be marked for destruction, because it's worthless, right? It can't. It's just going to be floating out there, not binding to anything. So, but the problem is, right? So that we don't want to bind to every single MHC. So now what we do is we have an MHC with self peptides on it. But it can't, if it binds it too tightly, right? We don't want it to activate every cell that displays an MHC, right? Because every cell has it. If it binds to a self protein, it can't. We don't want that out in the body causing havoc. And so, if that happens, then those those T cells are destroyed, stuff like that, because because we don't want those going out. So this is what the negative selection. Only if you pass this part here by not binding, okay, can you get in there. So you have to bind an MHC, but not bind an MHC with these self proteins on there. Okay? So that's how you teach it. So again, these are in the thymus usually. So that's what the T stands for, is thymus. Okay, so there are some autoimmune diseases, so you might want to know this list. And really, they're just a failure to recognize self-peptides. So what is happening is you attack healthy cells. So with, for type 1 diabetes, it's attacking the pancreas. Psoriasis, it's attacking the skin cells. Rheumatoid arthritis, it's the joints. Multiple sclerosis, it's the neuron, the coating, the sheath of the I don't remember what lupus is, but I know it's so how do vaccines work? Well, what it does is after you've activated your B cells, right? So those stay active, right? So and on the lookout for a secondary response, so in case for a secondary infection. And so the idea is if you have the B memory cells, you won't get that disease twice. So the way the vaccines work, right, is you actually give the infection initially, but in a way that it doesn't make you sick. So it can help you build up the T cell, or build up these B memory cells, so that you can launch the antibodies, the adaptive immune system, very, very quickly. So you've got the innate immune system that's holding things down, and you can come in with the adaptive one within like 2 to 3 days, something like that. And so that should help prevent a full scale infection. And so you won't get the disease a second time, something like that. And so you're tricking the body into making the B memory cells before you actually get sick the first time. So that's how that works. Okay, that was a lot. Some of that. So any questions?